Hello, I'm Ali Nikakhtar, your host at DystroBlog, a space for conversation and awareness about neuromuscular disease. Today, I want to talk with you about one of the most important breakthroughs in genetic therapies over the recent decades, the progress toward AAV-mediated gene therapy for Duchenne Muscular Dystrophy. Now, I've already shared a few videos on this topic on this channel, along with several educational posts on the website nikilab.net. Just a few days ago, I came across a valuable review article authored by three distinguished scientists, Jeffrey S. Chamberlain, Nicholas E. Bengtsson, and Tasfaout. All three are affiliated with the University of Washington Schools of Medicine in Seattle. I thought it would be worthwhile to share their insights here with you. Let's start with the big picture view. Duchenne Muscular Dystrophy, or DMD, is a severe and progressive genetic disorder that affects young boys. The disease is caused by mutations in the dystrophin gene. This gene is massive, about 2.2 million base pairs, and its sheer size not only makes it more valuable to mutation, but also makes therapeutic development incredibly complex. Over the past few decades, the central idea has been to deliver a healthy copy of the gene into muscle cells. But because of the full dystrophin gene is far too large to fit inside an AAV vector, researchers have designed a smaller version of it, like mini-dystrophin or micro-dystrophin. Why is that? Because the studies have shown that even when large portion of the dystrophin protein are missing, as we see in milder cases like Becker muscular dystrophy, it can still retain considerable functionality. That finding laid the groundwork for micro-dystrophin constructs, which are smaller but still offer meaningful protective effects. AAV vectors emerge as the best delivery option because of their relatively low immunogenicity and capacity for long-term expression in the muscle tissue. But their packaging limit is around 4.7 kilobases, meaning we can only fit a micro-dystrophin construct inside. To complement that, researchers also developed muscle-specific promoters like MCK and MHCK, which limit expression to muscle tissue and thereby reduce immune complications. The next step was systematic delivery, making sure gene expression reached all muscles throughout the body, including the heart. Achieving this requires high vector doses, careful control of injection parameters, and use of optimized serotypes like AAV9 or RH74, all of which help achieve broader biodistribution. Now, when it comes to clinical trial, 6 companies entered the field, including Sarepta, Pfizer, Solid Biosciences, Genethon, and REGENXBIO. Sarepta Therapeutics, in particular, is a familiar name to many families affected by muscular dystrophy. This US-based company was the first to receive FDA approval in 2024 for DMD gene therapy. However, while the results were good, they weren't outstanding in most cases, because dystrophin expression ranged from 20% to 18% of muscle fibers, and the clinical benefits remain modest. One major focus of the article was the immune responses triggered by the micro-dystrophin constructs or the AAV vector itself. Some patients experience serious adverse events like thrombotic microangiopathy or TMA or liver failure. That said, efforts to use muscle-specific promoters, reduce doses, and redesign micro-dystrophin constructs to eliminate immunogenic epitopes are making real progress in controlling these reactions. Another major leap forward has been development of next-generation vector called myotropic AAVs. These are engineered to target muscle tissue exclusively while reducing liver uptake. Early results from solid biosciences have shown that with these vectors, it's possible to reach much higher expression levels, even up to 135% of normal dystrophin in some patients, and with lower doses. But the big question remains: how can we deliver the full-length dystrophin gene to muscle? The answer lies in dual or triple vector approaches using transsplicing, ribosome-based methods, or split intent protein strategies. In these methods, different parts of the gene are delivered separately and then reassembled within the cell to produce a larger functional protein. These approaches are still in the animal testing phase, but the results are promising. Alongside these strategies, gene editing is emerging as a serious contender, whether through CRISPR-Cas9, base editing, or prime editing. These technologies have the potential to directly repair the patient's defective gene in its native location. However, challenges such as safety, delivery to muscle stem cells, and tight control over the timing of editor expression still needs to be resolved. In the end, the path toward treating DMD with AAV has brought us to a place where effective therapy seems truly within reach. But there's not there yet. We still need better vectors, safety delivery systems, and perhaps a combination of gene therapy and gene editing. The future is bright, but the journey continues. If you found this topic as engaging as I did, you can find me on LinkedIn, Twitter, YouTube, and of course, my website, nikilab.net, a hub for deeper insights and awareness. Do check it out. And remember, my dream is to see a smile on your lips.